Welcome to this short video where we're going to be looking at the National 5 topic, DNA and the production of proteins. It's going to be a relatively short video. We're going to go over the main points of the topic, and hopefully at the end of it you will have learned the difference between your adenine and your thymine, or your cytosine or your guanine. My name's Stuart White. I'm an experienced biology teacher. I've been teaching for over 20 years and I'm going to go through this whole topic with you just now. So first of all, we need to know that DNA is a double-stranded helix, okay? And we can see that in this diagram. We've got one strand here, and we've got the other strand here, okay? And they form a helix shape, a twisted ladder, okay? Within that twisted ladder, within that double helix, we have complementary base pairs, okay? Now in DNA, there are 4 bases: adenine, thymine, guanine, and cytosine, often abbreviated to the single letters A, T, G, and C. Now, the word complementary up here, complementary means that one goes with the other. So in this case, A goes with T, adenine goes with thymine, and G goes with C, so guanine goes with cytosine. And the handy way to remember this is to remember that you have apples in the tree, A with T, and that your car goes in the garage. Okay, so C with G, the car in the garage, okay? If we look at the diagram here, we can see that complementary base pairing happening in the DNA. Wherever we've got an A, we've got a T opposite. Wherever we've got a G, we've got a C opposite, okay? T with A, C with G, C with G, T with A, C with G. Okay, and if you remember that complementary base pairing rule, it's really hard to go wrong in these questions on DNA. Okay, so we need to remember this complementary base pairing rule. Now, DNA carries the genetic information for making proteins. Okay, all of the proteins in our body, our hair, our muscle, our tissues, our organs, etc., are all made by reading a genetic code from our DNA to then make proteins in our ribosome. Now, on our DNA, we have small sections, okay, small sections of our DNA called genes, and each of those genes has the code to make a particular protein. Okay, so a gene is a section of the DNA which codes for a particular protein. And how does that work? Well, when we read the base sequence of our DNA, okay, here's just a random sequence here of A, C, G, TAG, CAT. That base sequence, the order of those bases, determines the amino acid sequence, okay? These are amino acids, we've just randomly labelled them 1, 2, and 3 here, but you can see they are different amino acids because they are coded for by different bases, okay? And we tend to code for amino acids in base triplets here, okay? So in 3 bases, A, C, and G codes for amino acid 1, T, A, and G codes for amino acid 2, and C, A, and T codes for amino acid 3. Now, amino acids are the building blocks of protein. They are what protein are made of. Okay, so if you imagine little Lego blocks, the amino acids are the individual blocks, but then the model, the building, the airplane, whatever you make with the Lego, is the protein. Okay, so amino acids are the units or the building blocks of protein. So then that amino acid sequence will determine which type of protein our cell is going to produce. Okay, so the base sequence determines the amino acid sequence, which then determines the protein that's going to be produced in the end. Now, how does this work within our cells? Well, the DNA, that base sequence, that is stored in the nucleus, okay? We know that the DNA is kept in the nucleus. It's nice and safe in there. Those instructions in our DNA are vital for us to continue producing cells which can make their own proteins and can carry out all of the cell's activities. If that DNA was to be damaged, that would be a bad thing. So we don't just send it out from the nucleus. Instead, what we do is we take a copy of that section of the DNA that codes for the protein. Okay, that complementary copy of DNA is a molecule called mRNA, or messenger RNA. Now, the good thing is the messenger RNA can leave the nucleus, okay, and it does, it leaves the nucleus and it travels to this little organelle here called the ribosome. Now, we know from the first topic that the ribosome is the site of protein synthesis, so this makes sense. Send a complementary copy of the DNA from the nucleus to the ribosome. And when that complementary copy arrives at the ribosome, it then has the instructions, the base sequence, for therefore making our amino acids, which will then join together to form our protein, okay? So messenger RNA is a molecule which carries a complementary copy of the DNA from the nucleus to the ribosome to assemble proteins from amino acids. Okay folks, hope that was useful as a little recap to that topic. Let me know if you've got any questions or if you're still not sure about anything. But for now folks, class dismissed.